Chapter 14 it's about the brain and the cranial nerves. Brain has a weight of about 3.5 pounds in men and a little bit less in females. And the brain is subdivided into three major regions. The cerebrum, which makes 83% of the brain volume. And then within the cerebrum we have the cerebral hemispheres, Gyri and Sulci, and a longitudinal fissure that divides the cerebrum into two hemispheres. And then we have an inner connection that is called the corpus callosum that connects these two cerebral hemispheres. The other region of the brain is the cerebellum. And the cerebellum, although it's small in comparison to the brain, to the cerebrum, it has 50% of the neurons. And it's located within the occipital bone in the posterior cranial fossa. Brainstem is the portion of the brain that remains after the cerebrum and cerebellum are removed. Within the brainstem we have the Diencephalon, Midbrain, Pons, and Medulla Oblongata. So this is the lateral view of the brain, and then this area will be anterior, this will be posterior. This directionality is also called rostral, while this is called caudal. So in the surface of the brain you can see, or the cerebrum, which is this, you can see these convolutions that we call gyri, and then we have some indentations in between the gyri that we call sulcus. In the surface of the cerebrum we have deeper sulcus that has a specific name, like in the case of the central sulcus here or the lateral sulcus in here. The cerebrum has different subdivisions, like the temporal lobe in this case, and we will see the others in a few slides. And in here we have the cerebellum, and then the brainstem with its different components, and of course below here is the spinal cord. So this is the view of the three major portions of the brain looking medially. So this is the brainstem, this is the cerebrum, and this is the cerebellum. Longitudinal fissure, it's a deep groove that you can see from the superior portion of the cerebrum that separates the cerebral hemispheres into left and right. We have within the surface of the cerebrum these thick folds or convolutions or turns that we call gyri, and then we have divisions between the gyri that we call sulci, which are shallow grooves. Corpus callosum is a series of tracts or axons that connects left and right hemispheres, that it's located within the deep part of the longitudinal fissure. So this is the superior view of the cerebrum. So here you can see the longitudinal fissure separating the right from the left hemisphere. And then in this view you can see the frontal lobe, the parietal lobe in here, and the occipital lobe in here. This is an anterior view of the brain, so you can see of the cerebrum. You can see here the longitudinal fissure with the sulci and the gyri. So gyri are thick folds or convolutions or turns, while the sulci are shallow grooves. Here is the location of the corpus callosum, so this is the medial view. of the corpus callosum at the bottom of the cerebrum, interconnecting left and right hemispheres. As you can see here, this is a frontal section of the brain. So this will be right side, this is left, and this is the corpus callosum connecting both the right and left hemispheres. And you can see an MRI view of this structure in here and in here. And this is the superior view with a cross section, so you can see the corpus callosum, how it connects the two hemispheres of the cerebrum. For the cerebellum, it occupies the posterior cranial fossa in the inside of the occipital bone. Similarly as to the cerebrum, it also has gyri and sulci and fissures, but these are less deep. And this cerebellum is separated from the cerebrum by the transverse cerebral fissure. It makes around 10% of the volume of our brain, and then, as we said before, it has 50% of the neurons and of the brain, and then it is located in the posterior cranial fossa. Now, the brainstem will be the other part that remains of the brain if you remove the cerebrum and the cerebellum. Within the brainstem, we have subdivisions, the Diencephalon, Midbrain, Pons, and Medulla Oblongata from superior to inferior. So this is the location again of the cerebellum in the posterior cranial fossa, and then anterior to the cerebellum and inferior to the cerebrum, we have the brainstem. If you make a longitudinal section to see the medial view of the brain, you will be able to see the divisions of the brainstem. So this is the midbrain in here, this is the pons, and this is the medulla oblongata. Now within this medial view, you will be able also to see here the corpus callosum, and then the mid-cut of the cerebellum. And if you can see here, the cerebellum also has gyri and fissures. This gyri is more horizontally oriented than these ones that we have in the cerebrum. So, but they are called gyri, and then we have sulci. Now, another thing that you can see in this mid view is that under the corpus callosum, we have this region that we call the thalamus. And then posterior to the thalamus, we have the epithalamus with the gland that we call the pineal gland. And then we have here, anterior to the thalamus, the anterior commissure that, similarly as to the corpus callosum, also helps to connect the left and right hemispheres. And then below, we have the optic chiasma, and then we have the pituitary gland in here, the one that sits into the sella turcica of the sphenoid bone. And then just in between the brainstem and the cerebellum, we have this cavity that is called the fourth ventricle. And then here, in between the right and the left hemispheres, just underneath the corpus callosum, we have what we call the lateral ventricles, which are cavities that helps to bring the cerebrospinal fluid from the brain into the spinal cord. And just one note in here that is not labeled, but this white structure that you see here that runs partitioning at some point a little bit the cerebellum is called the arbor vitae, or the tree of life. So this is an actual view of the brain in the cadaver with all the structures I already mentioned to you. So here you see the lateral ventricle, And then in here we have the third ventricle, and then the fourth ventricle in here. So within the brain we have gray matter, that it is composed of bodies of neurons and dendrites and their synapses, and they are located within the cerebral cortex, in the cerebrum, and within the cerebellar cortex in the cerebellum. Now, within the base of the brain, we have what we call collections of neurons that, or body of neurons that we call nuclei. And if they're found within the base of the brain, these are called the basal nuclei. Now, we have also within the brain white matter that are bundles of axons that are located deep to the gray matter, both in the cortex and in the cerebellum. And they have axons or bundles of axons that connects the spinal cord to the different regions of the brain. And the color of the white matter comes from the presence of myelin around the axons. And this white matter is composed similarly as the spinal cord of bundles of axons that makes these tracks that goes up and down. So this is the location then of the gray matter in the cerebral cortex of the cerebrum, and white matter here, deeper. And then we have within this region several nuclei that you cannot see here right now in this coronal section, but this will make the basal ganglia, or nuclei, sorry. So our nervous system develops from the outer layer of the embryo that is called the ectoderm. And then early in the third week of development, within the midline of the embryo, we have a thickening of this ectoderm, and you form what we call the neural plate. And then this neural plate will form a groove that is going to be called the neural groove, and it will start going deep, and as you go deep, the neural plate starts coming closer to each other until you form a fusion between these two, and then sides of the neural grooves, and then you close the neural tube by almost week 4 of the life in utero. And then this neural tube will become filled with fluid that will later form the ventricles of the brain and the ependymal canal of the spinal cord. So this is the view of early embryo at 19 days. If you make a cross section and then you look at part of this cross section, you will see that this layer, the ectoderm, forms these neural crest cells that are going to form a line along the long axis of the embryo, and then in the middle of these two sides of the neural crest you will have the neural plate, and then that neural plate in orange is going to deepen by the next day, and then you start forming a fold, and then this bottom of the fold, which is going to be called the groove, is going to go a little bit more deeper, and as you're going deeper, these neural crests or folds are going to get closer together by around day 22, and then you start closing it by around day 24, and then you form this neural tube. And you can see how this folding is happening. So here you have on this longitudinal view this neural crest, and then how it starts closing here in the middle, and then that closure will follow superiorly and inferiorly, or rostrally and caudally. Now the neural crest is going to give rise to different structures in the adult, like the two inner meninges, the pia mater and the arachnoid, and also is going to form most of the peripheral nervous system and parts of the skeletal, integumentary, and endocrine systems. Now, by week 4, the neural tube will exist and is going to have 3 dilations or vesicles, and those will be within the cephalic end or anterior end or rostral end. So the most anterior dilation is going to be called prosencephalon or forebrain. And then we have a mid vesicle that is called the midbrain or mesencephalon. And then we have a tiny vesicle posterior to the midbrain that is called the hindbrain or rhombencephalon. By week 5, some of these vesicles will subdivide. So the forebrain, for instance, is going to divide into 2. So it's going to form the telencephalon, which is going to become the cerebral hemispheres, and then it's going to divide into the diencephalon, that is going to have the optic vesicles that will form a retina, and then the midbrain is not going to divide, so it's going to stay as mesencephalon, while the hindbrain is going to divide into 2 vesicles, the metencephalon and myelencephalon. Myelencephalon is going to form the spinal cord and also the rhombencephalon in here. So here is the lateral view of the embryo at week 4. So you can see here we have the spinal cord. This will be the rhombencephalon, mesencephalon, and prosencephalon. And then at this point, the prosencephalon starts to divide into the telencephalon in yellow and the optic vesicle in blue. And then in here in 5 weeks, you can see better this division. Mesencephalon remains undivided, and then this myelencephalon or rhombencephalon is going to divide into the metencephalon and myelencephalon, which is going to form the pons and the cerebellum, the metencephalon, and myelencephalon is going to form the medulla oblongata. Similarly, as for the spinal cord, the cerebrum or the brain is protected by 3 membranes, the meninges. And they have the same name from outer to inner: dura mater, arachnoid, and pia mater. The cranial dura mater has 2 layers. It has an outer layer that is in close contact with the skull bones. That's why it's called outer periosteal. And then the inner is going to be called meningeal because it's in close contact with the arachnoid. And then this meningeal is the one that is going to continue into the vertebral canal, forming the dural matter around the spinal cord. And then within these 2 layers, we have these blood vessels that helps to circulate the blood within the brain that are called the dural sinuses. Now, since the dura mater is in close contact with the cranial bones, we don't have epidural space within the brain. And then this dura mater doesn't have a direct attachment to the brain except around the foramen magnum, in the sella turcica, in this upper protrusion of the ethmoid that we call the crista galli, and at the sutures of the skull. And then this dura mater is going to fold in between different regions of the brain, like in between the cerebral hemispheres, where it's going to be called falx cerebri, in between the cerebellum and the cerebrum in what we call the tentorium cerebelli, and in between the right and the left hemispheres of the cerebellum into what we call the falx cerebelli. The arachnoid and the pia mater are similar to those of the spinal cord, so you will have extensions of connective tissue from the arachnoid into the dura mater, and then you will have subarachnoid space that separates the pia mater from the arachnoid, and then the subdural space that separates the dura mater above in some places, and then the pia mater, still like the pia mater in the spinal cord, it covers directly the brain tissue, and then it goes deep in between the sulci. So here is the view of these different layers or meninges. So we have here the dura mater with the periosteal layer and meningeal layer. Remember, we don't have here epidural space. And then in between these two, the periosteal and meningeal layer, we have these dural sinuses that has some names in some regions, like in this case, superior sagittal sinus. And then in here we have the arachnoid, and then in here we have the pia. And then the arachnoid has some protrusions that we call the arachnoid granulations that helps to absorb the cerebrospinal fluid as it is produced. And then some extensions of the dura mater, like this one that goes in between the cerebral hemispheres, is called the falx cerebri. Meningitis is inflammation of the meninges. It happens at any age, but it's more serious during infancy and childhood because the brain is developing, especially if it happens between 3 months or 2 years of age. It can be caused by bacteria, viruses, as I told you in the key point from last chapter. And these bacterial viruses can enter into the CNS by the way of the nose and throat. And the two meninges that are mostly affected will be pia mater and arachnoid because they have less thickness than the dura mater. And then meningitis, it can cause so much damage as enlargement of the ventricles because you have overproduction of the CSF, you can have inflammation, you can even have hemorrhage. And some signs of having meningitis is having high fever, having a stiffness of your neck, being drowsy, having an intense headache, including vomiting, what we call projectile vomiting. And if it's not diagnosed and treated properly, it can lead up to coma and death within hours of onset. And to diagnose meningitis, you have to obtain a sample of CSF by lumbar puncture in what we call spinal tap by introducing a needle in the subarachnoid space. Now, CSF is produced by choroid plexus, which is blood capillaries at the floor of the openings inside the cerebrum that we call the ventricles. Now, as this CSF is produced, it runs along the different areas of the brain and then the spinal cord, and it's supposed to be absorbed by the arachnoid granulations. And also, we have within the middle part of the spinal cord these ependymal cells that lines the ventricles and covers the cerebral plexus and also the ependymal canal in the spinal cord that produces the CSF actually. Now, the ventricles are 4. We have 2 lateral, each on the deep part of the right and left cerebral hemispheres. And then we have a pore that connects these lateral ventricles to the third ventricle that we call interventricular foramen. And then the third ventricle, which is located just underneath the corpus callosum, will connect to the fourth ventricle that is in between the midbrain and the cerebellum through the cerebral aqueduct. And then the fourth ventricle, which is again located between the midbrain and the cerebellum, will connect to the central canal that runs through the spinal cord. CSF is a fluid that is clear. It doesn't have a color, and it fills the ventricles and canals of CNS. It helps to bathe the external surface of cells, and it is produced and absorbed with around half a liter per day. Now, if you have more production than absorption, you have accumulation of this CSF, your ventricles will enlarge and you will have what is called hydrocephalus. Now, the ependymal cells, in order to form the CSF, will filtrate the blood, and then it will secrete this fluid that it will have more sodium chloride than plasma, but it has less potassium, calcium, and glucose, and very little protein. So the CSF is continuously moving throughout the CNS, and it will help to nourish and protect our neurons. I already said that the CSF is reabsorbed by the arachnoid granulations of the arachnoid matter. So some functions of the CSF: it produces buoyancy, basically help us to have our brain floating within the skull. And then it also it provides chemical stability because it rinses away any metabolic wastes produced by the nervous tissue, and also helps to regulate the chemical environment. And it provides a source of protection because it prevents concussions in the brain hitting directly, or in the skull hitting directly the cerebral cortex. So here is the lateral and anterior view of the ventricles. So this is the lateral ventricles. This is the interventricular foramen connecting the lateral ventricles to the third ventricle. And then in here we have the fourth ventricle, that it is connected by the cerebral aqueduct to the third ventricle. And then here we have the ependymal central canal, and then these lateral apertures that are located on the sides of the fourth ventricle brings the CSF back into the subarachnoid space. An MRI of the lateral ventricles, you can see here. This is the third, and down here the fourth. This is a cross section of the brain, so you can see this space is the ventricles. So this is how the CSF flows. So from lateral to third to fourth to lateral apertures back into the subarachnoid space, and then from fourth, including going into the ependymal canal, and then down through the lateral apertures into the subarachnoid space in the spinal cord. Okay, so the brain is supplied by branches of the internal carotid artery, which is a branch of the common carotid, which is a branch of the aorta. The brain is only 2% of our adult body weight, but it receives 15% of the blood supply, around 750 milliliters per minute. Why is this? Well, because we have many neurons, and our neurons have an important function to make us move our muscles, to think, to move our viscerae, etc. So the neurons, is they're very active, they require a lot of ATP, and all of those nutrients that makes ATP, like glucose, is brought into the neurons by the circulatory system through the blood. And then our neurons also require a lot of oxygen, and that oxygen is very important, since if you interrupt the blood supply into the brain by 10 seconds, you might have loss of consciousness. That's okay, but if you lose your cerebral circulation for 1 or 2 minutes, you start having severe impairment of the neuronal function. And if you go beyond 4 minutes, you have irreversible damage. So now another branch of the circulatory system, or another blood vessel that provides blood supply to the brain, will be the basilar arteries, that are branches of the vertebral arteries that comes from the cervical area. So we have these posterior cerebral arteries that are branches of the basilar artery, which is coming somewhere in here. And then we have here the posterior inferior cerebellar arteries that still are branches of this basilar artery. And then we have the other branches coming from the internal carotid, which is this, that provides the anterior and communicating arteries, the middle cerebral arteries, etc. All of them, they provide blood supply to the brain into what we call the circle of Willis, because all of these branches, anterior and posterior, make like a cycle, and they run around the pituitary gland. Now, the brain has to be protected by a special mechanism, and we have the brain barrier system that regulates what substances can go into the bloodstream and then into the brain, and what cannot go. So we have 2 points of entry that must be guarded: the blood capillaries throughout the brain tissue and the capillaries of the choroid plexus. So we have the first barrier, which is the blood-brain barrier. And then this will protect the blood capillaries throughout the brain tissue. And these capillaries has a special connection between the endothelial cells, or the cells of the capillaries. These connections are very, very strong, and we call them tight junctions. And they form this double layer in which you have the tight junctions of the endothelial cells and the astrocytes that surrounds them, with extensions of their cytoplasm that we call perivascular feet. And these extensions can open and close according to what it needs to go into the brain and what it shouldn't. So if something is important, the perivascular feet of the astrocytes will open, so whatever is coming off from the capillary walls through the tight junctions can go into the brain. So this blood-brain barrier will exclude harmful substances from passing into the brain, while allowing the necessary ones to enter. Now, we also have the blood CSF barrier, in which you protect the brain at the choroid plexus. So still you have tight junctions between the ependymal cells of the choroid plexus, and this will be important to allow the exchange between the brain tissue and the CSF. The brain barrier will be permeable to water, glucose, and lipid-soluble substances like oxygen, carbon dioxide, alcohol, caffeine, nicotine, and anesthetics, and it will be slightly permeable to ions like sodium, potassium chloride, and waste products of urea and creatinine. So the brain barrier system, then it has this problem. So it will be an obstacle for delivering medications like antibiotics and cancer drugs. Now, when you have trauma or infection or inflammation that damage the blood-brain barrier system, you will have opening between these cells that protects the brain, and then this opening will be used by microorganisms, and these microorganisms will go in, and then they produce infections. Okay, now let's talk about the medulla oblongata. So the medulla oblongata, it comes from the menincephalon, if you remember. And it will, the medulla oblongata starts at the foramen magnum, and it will extend rostrally around 1 inch, and it will end at a groove below the pons. And it will be slightly wider than the spinal cord. So here is the location of the medulla oblongata. It starts here at the foramen magnum and ends here at this groove where the pons is located. This anterior protrusion of the midbrain, of the brainstem, is called the pons. Anteriorly, the medulla oblongata is characterized by having this crossing of these nerves that are coming from the cerebral cortex that we already talked about, that are called the pyramids. And then these pyramids will be separated by the anterior median fissure. Now, within the medulla oblongata, we also have the emerging of cranial nerves, like the cranial nerve number 8 in part, which is the vestibulocochlear nerve, cranial nerve number 9, which is called glossopharyngeal, cranial nerve number 10, which is called vagus nerve, and cranial nerve number 12, which is called hypoglossal nerve. Now, within this medulla oblongata, we also have on the sides some protrusions that we call the olives that are lateral to the pyramids. And we also have these gracile and cuneate fasciculi of the spinal cord that continues as the posterior ridges on the posterior part of the medulla. So here is the anterior view of the medulla. So in this part that you see here, you have the pyramids, the crossing over of the nerves that comes from the cerebral cortex. And then this will be the pyramids as well in here. So this is the pyramidal decussation. And then in here, we have the emerging of cranial nerve number 12, or hypoglossal. And then within this part, we have spinal nerves, and we have cranial nerve number 11 emerging from here. And also in here we have the emerging of cranial nerve number 9, which is this one in here. And this part in here is called anterior median fissure. Dorsolateral view of the medulla oblongata. These protrusions are called the olives. And then in here we have cuneate fasciculus, and then gracile fasciculus in here. So all the ascending and descending fibers that connects the brain and the spinal cord has to pass through the medulla. And within the medulla, we have second order neurons of the gracile and cuneate fasciculi. If you remember from the previous chapter, we said that these neurons bring information about vibration, discrimination of touch, and proprioception. And then the fibers of these second order neurons will decussate and form a little band that we call the medial lemniscus. And the pyramids again bring down the fibers of the corticospinal tracts that carries these signals to contract our skeletal muscles. And then within the lateral portions of the medulla, we have these olives that has this olivary nucleus that helps to bring information into the cerebellum. And then as well, we have in here this loose network of nuclei that are called the reticular formation that extends from the medulla into the pons and the midbrain. And within this reticular formation, we have cardiovascular centers or cardiac and vasomotor centers and respiratory centers. So this is a cross section of the medulla oblongata. So in here we have the olive, and here we have the inferior olivary nucleus that brings this information into the cerebellum. And then we have the pyramids of the medulla that crosses these corticospinal tracts. And then we have here this medial lemniscus that is the connection of the fasciculus of the gracile fasciculus and the cuneate fasciculus. And we have here their nucleus. And then here we have the nucleus of the hypoglossal nerve, just anterior to the fourth ventricle. And then we have the nucleus of the vagus nerve. Hypoglossal nerve helps us to move our tongue. Vagus nerve helps to control several viscerae within the thoracic and the abdominal cavities. And then here we have the reticular formation that has these cardiac vasomotor centers. And then in here we have the tectospinal tract. So the pons is this anterior bulge of the brainstem that is anterior and superior to the medulla. This one comes from the metencephalon, and in here within the pons we have some structures that are called the cerebellar peduncles that helps to connect the pons to the cerebellum. Within these cerebellar peduncles we have ascending sensory tracts, descending motor tracts, and pathways that comes in and out of the cerebellum. Within the pons we have cranial nerves 5, 6, 7, and 8, or trigeminal, the facial nerve, which is number 7, abducens, which is 6, and 8, which is vestibulocochlear. This pons has roles in hearing, equilibrium, taste, facial sensations. And it has motor roles like eye movement, facial expressions, chewing, swallowing, urination, and secretion of saliva and tears, because these cranial nerves provide all this type of information. And then as well as in the medulla, we have reticular formation in the pons that has, in this case, functions for sleeping, for breathing, and for posture. So here is the location of the pons as well, or again, above the medulla oblongata. So it's superior and anterior. And then if you make a cross section of the pons, you will be able to see these fascicles, longitudinal transfer fascicles, that brings information from the medulla into the midbrain. And then we have here the superior middle cerebellar peduncles that helps to connect the midbrain and the pons to the cerebellum. And then in here we have the nucleus for the trigeminal nerve, and then here we have the medial lemniscus, and we also have here the other nuclei for the other nerves like 6, 7, and 8, but they are not shown in here because they are a little bit deeper. Now the mesencephalon becomes one mature brain structure, the midbrain. And then we have this mesencephalon that has the cerebral aqueduct that is surrounded by the gray matter that is involved in controlling pain. Now, within this mesencephalon or the midbrain, we have continuations of the medial lemniscus and reticular formation. And here we have the motor nuclei of cranial nerves number 3 or oculomotor and cranial nerve number 4, which is trochlear nerve. Within the mesencephalon, we have the tectum, which is a structure that looks like a roof, and it's posterior to the cerebral aqueduct. Within the tectum, we have a structure that is called the corpora quadrigemina. It has this superior colliculi or upper pair that it is important for movement of the eyes, so you can have visual attention, you can track moving objects, and you can have some reflexes. Now, within the lower pair of the corpora quadrigemina, that is called inferior colliculi, you have signals coming from the inner ear, so you can have reflective movements when you receive, for instance, information on one side of your body, as if it's coming a loud sound, and then you can turn to that side. So now within this area we have the cerebral peduncles that are two anterior stalks of the midbrain that will anchor the cerebrum to the brainstem. And then within the peduncles we have three parts that are called the substantia nigra, tegmentum, and cerebral crus or crossing. So the tegmentum, it has a nucleus that we call the red nucleus. And in here, it's called red nucleus because you have a lot of blood vessels. And this tegmentum also has these connections that runs to and from the cerebellum for motor control. Substantia nigra, it's a nucleus that it has melanin in it, and this is an area where we produce dopamine. This dopamine helps to inhibit unwanted body movement, and when someone doesn't produce dopamine, is going to develop what we call Parkinson's disease, and the person who has uncontrolled body movements or tremors. Cerebral crus is a crossing of bundles of nerve fibers that connects the cerebrum to the pons, and it carries corticospinal tracts. So here is the midbrain. So here's the cerebral aqueduct. You have the corpora quadrigemina here with the superior colliculus in here, and then in here you have reticular formation, and then you have the oculomotor or cranial nerve number 3, the nucleus for the oculomotor nerve, and then in here we have the medial lemniscus, red nucleus, and then we have here the substantia nigra, the one that produces dopamine, and then in here we have the cerebral crus. Reticular formation, again, it's a loose web of gray matter that runs from the medulla oblongata into the pons and then into the midbrain. It has many connections into the cerebrum, and it has more than 100 small neural networks without distinct boundaries. So it forms like a net. That's why it's called reticular. Ret means net. So here is the reticular formation that starts at the medulla, goes into the pons, and then into the midbrain. And it sends information to everywhere. So it will receive auditory input, visual input, and then sensory input, and then it will send back signals to the spinal cord. So functions of the reticular formation, you have somatic motor control, so you help us to adjust the tension in our muscles so we can maintain a specific tone and balance, so we don't fall, especially during certain body movements. And then also it relays the signals from the eyes and the ears to the cerebellum, so we can keep producing this balance, and then we can have motor coordination. And also I will send this information from the eyes, so we can track and fixate over objects, and then it will also generate some patterns that are called central patterns that helps to maintain rhythmically signals into the muscles of breathing and swallowing. Also within the reticular formation, we have centers that helps to control responses by the cardiac muscle and also by our circulatory system. And we have also modulation of the pain, and we have these analgesic pathways that helps to block transmission of pain signals. A very important function of the reticular formation is the controlling of sleep and consciousness. So it will help us to stay alert and to fall asleep when we're required to go and fall asleep. And whenever someone has or goes into an accident, like an automobile accident, and the person has whiplash, it can break part of this reticular formation during this whiplash, and then the person might fall into coma, and this can lead to irreversible coma. Now, sometimes the information that we receive within the reticular formation, it can lead to modulation of the activity of the cerebral cortex, so we can habituate and to certain information, and we ignore repetitive inconsequential stimuli. Okay, so that was about the brainstem. Now for the cerebellum. So the cerebellum is the largest part of the hindbrain and is the second largest part of the brain. It has these two hemispheres, left and right, separated by the vermis, which is located all along the superior and posterior surface of the cerebellum. It has a cortex that is superficial, and it has these folds or folia, which is similar to the gyri. And then it has this branching that we call the arbor vitae, this branching of white matter. And then we have nuclei within the cerebellum. It has about 100 billion neurons, and these neurons are called Purkinje fibers or Purkinje cells. Now, the cerebellum connects to the brainstem by the three cerebellar peduncles. And the inferior peduncles will connect specifically to the medulla oblongata, so that most of the input that is coming from the spinal cord will enter the cerebellum. Now, the middle peduncles connects to the pons, and then it will receive the input from the rest of the brain, and the superior peduncles are connected to the midbrain, carries the cerebellar output. Okay, so this is the medial view of the cerebellum. This is the superior view of the cerebellum. So this is anterior, this is posterior. So things that you can see here in the medial view will be the cortex, all of this here, and then the white matter, which is the arbor vitae. And then here in the superior view, you can see the right and the left hemispheres, and then the vermis separating them, and then the foldings or the gyri that are called the folia. So functions of the cerebellum. So it's motor coordination and locomotor ability. Also, it is shown recently that it might have sensory, linguistic, and emotional functions that are non-related to motor functions. So it helps to compare textures of object, perceive space, recognize object from different views, and then keeping judgment between tapping and a tapping rhythm, and also helps direct eye movements that compensates for head movements so that the gaze stays on a fixed object as you move the head. And also help us to judge pitch of tones and distinguish between similar spoken words. And help us also to associate verbal association tasks, planning, scheduling, and having emotional control, as it seems like many hyperactive children has smaller cerebellums. Okay, now let's talk about the forebrain. So the forebrain has the encephalon that has the third ventricle, and the telencephalon that is the one that it will develop into the cerebrum. So this is the forebrain with the telencephalon in here, the encephalon in here. So the diencephalon has the thalamus, hypothalamus, and epithalamus. So go back, or hold on. So here the diencephalon has the thalamus, hypothalamus, and epithalamus in here. So the thalamus, it's ovoid shape, and it's located on each side of the brain and constitutes about 4/5 of the encephalon. The two thalami are joined by the intermediate mass, and within the thalamus we have 22 nuclei that functions as 5 major functional groups. So this is the thalamus. This is the location of the thalamus. So it has an ovoid shape, and it has these functional groups: the anterior, medial, the lateral group, the posterior group, and the ventral group in here. Functions of each of these: the anterior group helps to form part of the limbic system, which is in charge of emotional control and memory. Medial group is emotional output given into the prefrontal cortex and also awareness of emotions. So I guess people who doesn't have conscience, their medial group doesn't function because they're not aware. For the ventral group, somatostatic output to the postcentral gyrus. And then it also sends signals to the cerebellum, to the basal nuclei of the motor cortex. And then lateral group, it has similar functions as for the ventral group, but it also contributes to emotional function of the limbic system, similar to the anterior group. And the posterior group help us to relay signals into the occipital lobe, like visual signals, and then auditory signals into the temporal lobe via the geniculate nucleus. The thalamus is a gateway to cerebral cortex, so you can filter the information in there, whatever it needs to go to cerebral cortex and whatever it doesn't need to go. For instance, if you are hearing lecture and whatever the lecturer is going to say, it's important, but you also want to go to pee, you might ignore going to pee because you know that that information that probably your lecturer is telling is not going to be repeated. So you filter that information of going to the restroom. So you keep it in the thalamus, and then you pay attention by sending that information into the cerebral cortex. Hypothalamus is below the thalamus, and it forms the walls and the floor of the third ventricle. It will extend anteriorly to the optic chiasma and posteriorly to the mammillary bodies, And then this hypothalamus has different functions, including relaying signals into the limbic system to the thalamus, and then connecting the pituitary gland to the hypothalamus. So, some of the functions of the hypothalamus is to perform homeostatic mechanisms. For instance, we secrete hormones that are important for regulating growth, metabolism, reproduction, and stress responses. Also, it helps to send these hormones like oxytocin and ADH that are going to be important for contraction of the smooth muscle cells within the uterus during labor, and also contraction of the smooth muscle cells around the glands of the breast in females during lactation. And then this ADH is going to, antidiuretic hormone, is going to help us to absorb water when we are dehydrated. Within the hypothalamus, we also has these centers of the ANS that can change the heart rate, blood pressure, the secretion of gastrointestinal tract glands, and also the motility of the gastrointestinal tract. We also have a thermoregulatory center that controls the body temperature. And then we have centers that controls appetite and water intake. So we have the third center there, and then the hunger and satiety center. And we also have this nucleus, the suprachiasmatic nucleus, that controls the circadian rhythm, so that we can stay awake during the day and fall asleep during night. And we also have these mammillary nuclei that help us to retain information. And we also have emotional behavior and sexual response centers that controls anger, aggression, fear, pleasure, contentment, and sexual drive. So this is the hypothalamus with all its centers. And then we have this part that is called the stalk of the pituitary gland that connects this preoptic nucleus that secretes ADH and supraoptic nucleus that, supraoptic nucleus produces ADH, and then we have the preoptic nucleus that secretes oxytocin. So you can release that into the pituitary gland, and then you can release them into the circulation. Sorry again, paraventricular nucleus is the one that produces oxytocin. Hypothalamus is a very small part of the diencephalon, and is composed of pineal gland, which is part of the endocrine system. It also has a structure that's called habenula. This will relay the information from the limbic system into the midbrain, and is located over the roof of the third ventricle. So this is the location of the epithalamus. So here's the habenula, and here it is the pineal gland. Okay, for the cerebral, this is the most large part of the brain. This is the area where we have sensory perception. We have regions of the cerebellum that are related to storing memory as well. We process thought there, judgment, voluntary motor actions. And then the cerebrum is subdivided into cerebral hemispheres by the longitudinal fissure that it is connected, the cerebral hemispheres, by the corpus callosum. In the surface of the cerebrum, we have Gyri and Sulci that increases the surface area, so we can have many neurons within the cortex. Now, the cerebrum is subdivided into different lobes, 5 major lobes according to the bone of the skull that they are connected to or in contact with. So this will be the right and the left hemispheres in the superior view. So here we have the frontal lobe, here we have the parietal lobe in here, and then in here we have temporal lobe and the occipital lobe in here. Now, separating the temporal lobe from the parietal lobe and the temporal lobe from the frontal lobe, we have this lateral sulcus, and then we have here the central sulcus that separates the frontal lobe from the parietal lobe. Functions of the frontal lobe: control voluntary movements, so it's having voluntary motor functions. It also motivates us. It's in charge of planning and foreseeing, so what will be the consequences. Help us also to store some memory, control of emotions, mood, social judgment, and aggression. Parietal lobe functions will be to integrate general senses, also has taste centers, and process some visual information. This one is subdivided by the central sulcus into the parietal occipital sulcus and the parietal lobe. Occipital lobe is in charge of detecting vision. So we have the primary visual center of the brain in there. Temporal lobe, it's in charge of hearing, smell, learning, memory, and some aspect of vision and emotion. And then in between the temporal lobe and the parietal, just deep to the lateral sulcus, we have the insula, which is another lobe. And then this one is important for understanding spoken language, taste, and then also integrating information from visceral receptors. So this is the location of the insula. And then in here we have the central sulcus that subdivides this area of the cerebrum into two important gyri: the precentral gyrus in the anterior portion of the central sulcus and postcentral gyrus in the posterior region of the central sulcus. And here it is, all the different functions of each of these lobes. The white matter again has bundles of nerve fibers that are myelinated and connects the different regions of the brain. So these fibers that connect the different of the regions of the brain are called projection tracts, commissural tracts, or association tracts. So the projection tracts extend vertically between higher and lower brain areas and the spinal cord. Example, we have the corticospinal tract. Commissural tracts runs from side to side of the brain, so connects the cerebral hemispheres. We have the corpus callosum, anterior and posterior commissures as an example of commissural tracts. And then association tracts connects the different lobes of the brain, and they are short fibers that connects a specific gyri within a lobe as well. So here is the location of the white matter and their different subdivisions. So we have here these projection tracts that goes from upper centers to lower centers. And then we have here the commissural tracts like the corpus callosum. Here again we have these projection tracts. And then we have in red these association tracts that connects the lobes or the sulci within the lobes, or sorry, the gyri within the lobes. Cerebral cortex is in charge of integrating the information that is coming into the cerebral cortex, and it will send this information through these tracks into the different regions of the brain or into the spinal cord, so that you can have more processing of the information or execution of commands. Now, the cerebral gray matter is found at the cerebral cortex, basal nuclei, and the limbic system. The cerebral cortex is having a thickness of 2 to 3 millimeters. It makes 40% of the brain mass, and it has between 14 to 16 billion neurons. And 90% of the human cerebral cortex, it is found in a region that is called the neocortex, that it has 6 layers. So if you magnify this part of the cerebral cortex, you can see all these 6 layers of the cerebral cortex. And in here you have these pyramidal-like neurons that are multipolar. It makes millions and millions of connections as a whole. Within the cerebral cortex, we not only have these pyramidal-like cells or pyramidal cells, but we also have these stellate cells that looks like stars. The stellate cells, they receive sensory input and process the information at the local level, while the pyramidal cells, they send output information and they connect with other parts of the CNS. Limbic system is made by different regions of the brain around the thalamus. They have an important function for processing emotion and learning. So we have subdivisions of this limbic system. So we have the cingulate gyrus, we have the hippocampus, and the amygdala. The limbic system connects through a loop of fiber tracks that follows somewhat a particular pattern, a circular pattern that forms feedback between the different areas of the limbic system. Now, those 3 components that we say, cingulate, gyrus, hippocampus, and amygdala, are not the only parts that influence the limbic system. The limbic system also can receive information from temporal lobe, from frontal lobe, and within the limbic system, we have structures that have both gratification and aversion. So gratification is sensations of pleasure or reward, while aversion will be sensations of fear and sorrow. So here are some of the components of the limbic system. All of these, like the orbitofrontal cortex, the cingulate gyrus, fornix in here, the thalamic nuclei, hippocampus in here, the amygdala in here, all form a complex system. The basal nuclei are located deep in the white matter, lateral to the thalamus. They receive input from substantia nigra and motor areas, and they help to control the movement of our muscles, our skeletal muscles. So whenever we have damage to the basal nuclei, we end up making gross movements. So within the basal area of the brain, then we have these basal nuclei that are called the corpus striatum, caudate, putamen, and globus pallidus. And then we have the lentiform nuclei that are formed by exclusively putamen and globus pallidus together. So here we have the thalamus, and on the side of the thalamus we have these basal nuclei. So this will be the globus pallidus that together with the putamen forms the lentiform nuclei, and then when they form a triad with the caudate nuclei, you form the corpus striatum. Again, they control the movement of our muscles, the skeletal muscles. So emotion are feelings and memories that are interacting between the prefrontal cortex and the diencephalon. Prefrontal cortex will be an area where we have judgment, intent, and control over expression of emotions. And then within these feelings, we have fear that arises from the hypothalamus and the amygdala. The amygdala will help because it will receive input information from various sensory systems, and it will help us to control this sensory information that will be playing a role in fear, food intake, sexual behavior, drawing attention, and one of the outputs of this amygdala will go into the hypothalamus to influence somatic and visceral motor systems. So when we, for instance, are fearful, our heart will increase the heart rate, the blood pressure will rise, and then you will have goosebumps, or you can even have vomiting. Now, within this amygdala, we also have output that goes into the prefrontal cortex that controls the expression of emotions. So we have the ability to express love, control anger, and overcome fear, hopefully. And then our behavior is shaped by learned associations between the different areas of the brain that includes stimuli, responses to them, and then reward or punishment. Now, sensation is received by the area of the cerebral cortex that is called primary sensory cortex that is located within the parietal lobe. And this primary sensory cortex can send information into some areas of the cerebral cortex that we call association areas. That's where you interpret that sensory information. And then part of these association areas will be the primary visual cortex that we have within the occipital lobe that help us to make sense of the visual stimuli. And we have multimodal association areas that integrates many perceptions of our surroundings, and we can make sense of what is going on. So here is the left cerebral cortex showing some functional regions of this cortex. So here we have the motor association area, the Broca area that help us to articulate words. We have the prefrontal cortex, the olfactory association area, Wernicke's area that also associated with understanding language, visual association area that receives input from our eyes, and then you process this area before putting it into the primary visual cortex. Then we have here the auditory association area and somatosensory area. So here, anterior to the central sulcus, we have the primary motor cortex. So we send command for our muscles, skeletal muscles, to move using this primary motor cortex. And then posterior to the central sulcus, we have this primary somatosensory cortex in which we receive sensory information from the different regions of our body, and we can receive information, sensory information like pain, temperature, touch into this area, and then with this somatosensory area, we interpret this information. Okay, so that's all about the cerebrum. Now for the special senses. They are limited to the head, and they employ relatively complex sense organs like vision. So vision is visual information that it is processed within the visual primary cortex area and also within the visual association area. And it's important so you can recognize faces and familiar objects. And for hearing, we have this primary auditory cortex that is located within the temporal lobe and the insula, and this auditory cortex that is located within the temporal lobe deep to the inferior, deep and inferior to the primary auditory cortex. This hearing cortex help us to recognize spoken words and a voice or sounds. Equilibrium is another special sense. It's dealing with signals from balance that are coming from the cerebellum and also from the inner ear. Now, we have an association cortex at the roof of the lateral sulcus next to the central sulcus that help us to make consciousness of our body movements and orientation space. The other two special senses are taste and smell. So we have the gustatory taste that sends these signals or process these signals in the primary gustatory cortex that is located within the postcentral gyrus of the parietal lobe and is anterior to the insula. The olfactory sense is processed by the primary olfactory cortex in the medial lobe located within the deeper portion of the temporal lobe and inferior portion of the frontal lobe in the insula. Now we have general senses that are called somesthetic or somatosensory or somatic. These will include receiving sensory information, general sensory information like touch, pressure, temperature, pain, and stretch. Now, the area of the body that process these general sense, or the area of the brain that process these general senses, will be the thalamus, also the cerebral cortex, in what we call the primary somesthetic cortex, and the association area, the somesthetic association area that is located within the postcentral gyrus in the roof of the lateral sulcus. That's where we make cognitive sense of the stimulus. Motor control is given anteriorly to the central sulcus in the premotor area of the frontal lobes. And this area will have neurons that will send this signal, the upper motor neurons, into the lower motor neurons so we can have the contraction of our muscles. Okay, so here we have the precentral gyrus ahead of the central sulcus. This precentral gyrus, together with the premotor area that is located here, help us to regulate motor movement. And then here in the postcentral gyrus that is located posterior to the central sulcus, we receive sensory information so we can be aware of different sensory modalities like pain, touch, temperature, etc. So this is the primary somesthetic cortex in the precentral gyrus. Now, within this precentral gyrus, we have different areas that controls movements of different parts of our body. So these different areas are represented by what we call the motor homunculus. And this motor homunculus will be a distorted look of all the different regions of our body because they are controlled by these neurons with different intensity. the more distorted that you see, or the bigger the area that you see, the more distorted that you see it, that means that that area of the body, it is controlled by, with much intensity, by more neurons, by the motor area. So again, within this primary motor cortex, we generate electric signals or action potentials that will control skeletal motor movement, skeletal muscle movement. So with this area, the one that it is anterior to the central gyrus, the precentral gyrus. So if you make a section of this precentral gyrus, and then you see it in here with this motor homunculus, this is the primary somatosensory cortex. Sorry, this is the postcentral gyrus representation. Let's go into the motor cortex, the one that we were talking about before. So this is the primary motor cortex. And if you see these different regions of the body, they are distorted. They have different sizes. And this is a coronal section of this primary motor cortex. So within this deep portion of the precentral gyrus, we move our toes. Then within this, we move our feet, our ankle in here, our leg in here, the knee in here, hip, etc. So everything below the wrist, all these different regions are moved by these different areas of the precentral gyrus. Now look at the hand. See how we have specific regions to move specific fingers. But see how the thumb, or finger number 1, it has this large size. Because this area, it controls with more intensity the movement of the thumb than any other area. And look at the face. The muscles of facial expression are controlled by this big region of this primary motor cortex. And look at the lips. And look at the tongue and the pharynx. The tongue, it has a big area dedicated for movement because it's very important for vocalization, salivation, masticating, and swallowing. Okay, now let's go back into the somatosensory cortex, the one that we use to feel sensations like pain, temperature, touch. Okay. Similarly as to the motor homunculus, this is the sensory homunculus, we have different regions that controls different that receives sensory information from different parts of the body. And again, look at the distortion of the different regions of the body. So we have big hands and big fingers because we receive a lot of sensory information. And then as well here for the face, we have a lot of sensory information coming from the face, but most from the tongue and the lips. And then in here, the bottom part of this postcentral gyrus, we have information also coming from abdominal viscerae. And look here, we have information coming from the genitalia. And again, you can see here in this view the insula, which help us to detect information like taste. Okay, now let's for the sensory homunculus. Let's see some information in here. So it's a diagram of the primary somatostatic cortex that resembles an upside-down sensory map of the contralateral side of the body. What this means is whatever you feel, like if you pinch your index finger, for instance, with a nail, your left index, you'll feel it with your right side of the brain, or you interpret it, okay? And then will be similarly for the motor pathway. So you will control your left side of the body with the right side of the brain. And everything is connected at some point to the cerebellum when it comes to the motor pathways. Okay, so your motor pathways bring information into the cerebellum so you can control your movements. Okay, now the last portion of this chapter is about the cranial nerves. So the cranial nerves exit from the cranium or enter into the cranium. So we have 12 pairs, and these cranial nerves are actually part of the peripheral nervous system, or the PNS. And when they exit to the brain, they exit through foramina, or when they enter to the brain, they enter through foramina or holes. And these cranial nerves, they help to control the movement of muscles of the head and the neck, or the function of glands or sense organs that are located within the head or the neck. Now, the cranial nerves have pathways. Some are motor, some are sensory. And most cranial nerves will carry fibers between the brainstem and ipsilateral receptors and effectors. Exception, optic nerve or cranial nerve number 2, in which half of the fibers of this nerve will cross and the other half will go ipsilateral. As well, the trochlear nerve, all the efferent fibers lead to a muscle on the contralateral eye. Trochlear nerve is cranial nerve number 4. Now, the classification of the cranial nerves according to the information that they process is called motor, sensory, or mixed. So sensory nerves, the only ones that they receive afferent information, are cranial nerve number 1, which is olfactory, the one that make us smell, cranial nerve number 2, or optic, the one that make us see, and cranial nerve number 8, the one that make us hear and maintain balance and equilibrium. Motor cranial nerves will be cranial nerve number 3, which is called oculomotor, cranial nerve number 4, which is trochlear, cranial nerve number 6, which is abducens. All these three, cranial nerve number 3, 4, and 6, innervate the eye. Cranial nerve number 11, which is called spinal, and cranial nerve number 12, which is hypoglossal. Cranial nerve number 11 innervate muscles like the trapezius that we have in the back. And then cranial nerve number 12 controls the movement of the muscles of the tongue. So when you stimulate muscle with these cranial nerves, you have also fibers that detects proprioception. Now we have mixed nerves like cranial nerve number 5 or trigeminal, cranial nerve number 7 or facial, cranial nerve number 9 or glossopharyngeal, and cranial nerve number 10 or vagus nerve. So they have sensory functions and motor functions that might be unrelated. Cranial nerve number 7 has a sensory role in taste and motor in facial expression. So as you see, these are unrelated. So this is a picture of olfactory nerve. Olfactory nerve starts with these fascicles of the olfactory nerve at the roof of our nasal cavity. These nerves penetrate the cribriform plate of the ethmoid, and then they connect to the olfactory bulb, which is going to extend axons that will form the olfactory tract, and then they will reach this olfactory cortex within the insula. Optic nerve number, cranial nerve number 2, or optic nerve. This will start at the retina of the eyes. So this is the inferior view of the eyes. This is a cross section of the eye. So we have the retina here that receives photons of light so we can see, and then it will extend axons to form the optic nerve. And this will be the optic nerve that it will cross anteriorly to the pituitary gland, part of the fibers, and the other fibers will go in the same side, ipsilateral. So cranial nerve number 2 and 1 are the only nerves of the cranial nerves that starts outside of the brain and then goes into the brain. Cranial nerve number 2, or optic nerve, enters through the optic canal of the sphenoid. Oculomotor, or cranial nerve number 3. This one innervates our iris, so we can have pupil dilation or contraction of the pupil, so we can open and close our pupil in protection for the light. If you go into a very bright room, your pupil will contract, so you don't have so much light coming to your retina, so you don't damage your optic nerve or cranial nerve number 1. And then this one, the oculomotor, also connects to different extrinsic muscles of the eye. So it has a superior and an inferior branch, and it has this ganglion that is called the celiac ganglion. So these are superior and inferior branches of the oculomotor nerve. Trochlear nerve, or cranial nerve number 4, innervates superior oblique muscle, which is an extraocular muscle. Trigeminal nerve, or cranial nerve number 5, is the thickest and largest cranial nerve. It has 3 branches: ophthalmic division, or B1, which is sensory. It has a maxillary division, or B2, which is sensory, and it has a mixed division, which is called mandibular, or B3. So here is the trigeminal nerve. It has this ganglion that is called the trigeminal ganglion. And it has these 3 branches: the ophthalmic, or B1, the maxillary, or B2, and the mandibular, or B3. And here is the dermatome for this trigeminal nerve. So we receive sensory information with B1 from above the nose and all this region next to the temple, and also from our eyes, the frontal region, part of the upper scalp. And then this is B2, which receives information from the lips up to the lower part of the nose, the cheek, the side of the temple is sensory information that we receive. And cranial nerve, B3, so this is the maxillary branch, this is the mandibular branch, which receives sensory information from the lower lip, the jaw, part of the anterior portion of our earlobe, and also the posterior and lateral region of the scalp in this area next to the temple. And this also has this motor innervation that help us to move all these muscles of mastication like temporalis, lateral pterygoid, medial pterygoid, masseter, and the digastric muscle underneath our jaw. So the inferior alveolar nerve receives sensory information from our teeth, superior alveolar nerve from the superior teeth. And if you have a toothache, you know that the cranial nerve number 5 is sending that information to the brain. Abducens, cranial nerve number 6, innervates lateral rectus muscle in here. The rest of the extraocular muscles are innervated by oculomotor. Facial nerve innervates the muscles of facial expression. That's the motor innervation. And then also innervates salivary glands like the submandibular and sublingual gland, also our tear or lacrimal glands or tear glands, and nasal and palatal glands. Sensory information will receive taste from the 2 anterior thirds of the tongue, and when you have damage to this nerve, you will have no sweet or salty sensation, and that's for the sensory portion. If you have damage to the motor portion, you won't be able to produce contraction of your muscles of facial expression. This is what we call Bell's palsy. And you test this by putting into the tongue of the person a sweet or salty substance or bitter substances. When you test the motor function is by asking the person to do some facial expressions like smiling, frowning, raising their eyebrows. So this is the distribution of facial nerve along the different regions of our face. So it has this pterygopalatine ganglion in here, geniculate ganglion in here, and then the submandibular ganglion in here to innervate our salivary glands, the 2 anterior thirds of the tongue, the nose, the palate, and the muscles of facial expression in here. Cranial nerve number 8, vestibulocochlear, has 2 subdivisions. It has this cochlear nerve and the vestibular nerve that merges. So this is the cochlear and this is the vestibular to form the vestibulocochlear. So we hear with the cochlear nerve, and we maintain balance with the vestibular branch of the vestibulocochlear nerve. Glossopharyngeal, cranial nerve number 9. It innervates the posterior third of the tongue and also innervates muscles of the pharynx like the pharyngeal constrictors. Also receives signals from the carotid sinus and innervates the parotid gland, the largest gland in our oral cavity. Vagus nerve is the longest cranial nerve. It innervates pharyngeal muscles, innervates parts of the larynx. It also receives signals from the carotid sinus, sends nerves to innervate the heart, the lungs, so you can decrease the heart rate. It sends fibers to abdominal viscera like the liver, the stomach, the gastrointestinal tract, so you increase the motility of the gastrointestinal tract. So vagus nerve or cranial nerve number 10 forms part of the parasympathetic system. Accessory nerve or cranial nerve number 11 or spinal accessory. It provides innervation to several structures here within the neck, including muscles like sternocleidomastoid muscle and the trapezius. Hypoglossal nerve or cranial nerve number 12. It innervates intrinsic muscles of our tongue, the muscles that are inside our tongue, and extrinsic muscles of our tongue like styloglossus, hyoglossus, etc. So this is the summary of the cranial nerves. So we have mixed nerves. We have those nerves that has motor and sensory function. We have purely sensory, like cranial nerve number 2, for instance, cranial nerve number 1, and we have motor nerves like accessory or cranial nerve number 12, which is the hypoglossal nerve. Okay, so let me then tell you the key points for this chapter. So again, bacteria, viruses, and traumatic brain injury, inflammation of the meninges, and the CSF flow disruption can cause meningitis. Now, the blood vessels that provides blood supply to the brain will be internal carotid arteries and vertebral arteries. Now, cerebellum. Functions of the cerebellum will be maintenance of balance and equilibrium by monitoring postural muscles and by making quick adjustments. It will receive input from the motor cortex and the basal ganglia. It will compare intended movements with the actual movements, and loss of motor coordination or jerky movements will be formed if the cerebellum is damaged. So again, loss of motor coordination, jerky movements will form if the cerebellum is damaged. So cranial nerve number 5, trigeminal nerve, is associated with tooth pain. Cranial nerve number 7 is associated with both hearing and equilibrium. So both statements are true, that cranial nerve number 8 is associated with both hearing and equilibrium, and cranial nerve number 5 is associated with tooth pain. Now, if a person can hear and understand individual words, but he cannot understand the meaning of a set of words or phrases in sentences, this person has damage to the general interpretative area. So when someone can understand and hear individual words, but he cannot make sense of sentences, he has problems or damage to the general interpretative area. While a damage to the occipital lobe can damage the sense of vision, a damage to the temporal bone will likely affect the sense of hearing and balance. So hearing and balance is related to damage to the temporal bone, while damage to the occipital bone will cause problems of vision. So this is then all that I have for this chapter. Again, hopefully you can see it all, and have a good day or have a good night. Bye-bye.